Namaskaram. On behalf of the Hindu Temple Society of North America, Flushing, New York, Sri VALLABHA GANAPATI DEVASTHANAM, I welcome all of you, our audience on Facebook and YouTube, to today's session of In Conversation with Pãtašala. It's third of our series, and today we'll focus on Let's Talk Science, science behind the COVID mRNA vaccine. We have students from the Ganeša Pãtašala presenting some basic concepts going on to the development of the vaccine. Our first two presenters will be Arshvita Parthasarathy, who will be focusing on the cell structure, which will be followed by Kavya Bhatt, who will talk about DNA and DNA replication.
Namaste and hello. My name is Arshvita Parthasarathy. Our topic today will be understanding the COVID-19 vaccine and its machinery. We will briefly discuss the topic of the cell structure. Table of contents. What are cells? An intro to what cells are. Where is it in the cell? Where are the organelles located in the cells and how do they look like? Glossary of terms used in the cell. Understanding the organelles and the membranes inside a cell and what their functions are. Closer look at the cell membrane. What are cells? Cells are the structural and basic unit of life. Every single organism consists of cells. They are the building blocks of living things. Cells convert various nutrients taken from the food that we consume and transforms the food to energy. Cells contain many different organelles, and each organelle in the cell has its own structure and function. The complete set of genetic material, known as the DNA, is found in the nucleus of a cell. The interior of a cell holds different organelles and is surrounded by a wall known as a cell membrane. We will also look at what the cell membrane is and what it does. This is important so we can understand how a virus can get into the cell and how the vaccine works with the cell and its machinery to fight off the virus. So these two pictures here depict the animal or human cell. Where is it in the cell? Let's start off with the cytoplasm. The cytoplasm is a thick, clear, jelly substance that fills in the cell. All the cell organelles are suspended in the cytoplasm. The nucleus. This contains the DNA, which is organized into chromosomes. But what are chromosomes, you might ask? Chromosomes are long strands of DNA. So the nucleus contains the genetic information needed and an organelle inside it known as a nucleolus. And this light green blob here is a nucleolus. Cell membrane. This supports and protects the cell from the external substances. It also controls the movement of certain substances in and out of the cell. This makes the cell membrane semi-permeable, meaning that it allows some substances in and blocks other from getting through. It's kind of like a guard for the cell. Nucleus. A closer look. The nucleus has its own membrane called the nuclear envelope or the nuclear membrane. This envelope is made up of two layers of membrane, an outer membrane and an inner membrane. There's a thin space between the two layers of the nuclear envelope, and this space is directly connected to the interior of another membranous ER organelle, the endoplasmic reticulum. There are holes in the nuclear envelope which control which substances enter and exit the nucleus. So these nuclear poles are the holes that control which substances enter and exit. Where is it in the cell? Now let me show you the different organelles suspended inside the cell. Ribosomes over here, the nucleus membrane, a membrane surrounding the nucleus. Inside it we have the nucleus, and inside that we have the nucleolus. The smooth ER, smooth endoplasmic reticulum, the lyosomes, the vacuole, the Golgi body or the Golgi apparatus, the mitochondria, and the rough ER. So what's the difference between these two endoplasmic reticulums? Well, the rough ER has ribosomes out on surface, giving it a rough appearance. Glossary terms of the cell organelles. Let's start off with the nucleolus. The nucleolus is a factor producing ribosomal RNA. There are no chromosomes in the nucleolus, meaning it does not contain any DNA. Ribosomes are small organelles where proteins are made. There are small particles that are full of RNA. They are molecular machines that can produce proteins by forming chains of amino acids. This process is called protein synthesis. Smooth endoplasmic reticulum makes a lot of different substances that the cell needs, like lipids, natural steroids, and carbohydrates. It is also involved in the transportation of some substances in the cell. Rough endoplasmic reticulum. The main difference between the rough and smooth ER is the presence of the ribosomes out on the surface. The ribosomes outside give the look of a rough appearance, and that is also how the rough ER got its name. The rough ER also plays a role in making proteins that are either packaged and sent to the Golgi body or the cell membrane. It also transports some materials throughout the cell. Glossary terms of the cell organelles continued. Golgi apparatus or Golgi body. This is made from a stack of membrane-bound bodies called a cisternae. It acts like the cell's post office, packaging proteins, carbohydrates, and lipid molecules into vesicles, which are small pouches, to deliver them to specific places. Lysosomes contain enzymes that can digest proteins and lipids. This helps to break down worn out and bad cell parts that are no longer needed. This is why they also have the nickname of suicide bags. Mitochondria mainly converts energy found in glucose and stores it in molecules called ATP. ATP is the main energy currency that the cell uses. The vacuole looks like a blob. It is a fluid-filled organelle that has a membrane surrounding it. It also stores food and water for the cell. Vacuoles also contain other waste materials which might be a threat to the cell, so the vacuole helps isolate them and remove them. A closer look at the cell membrane. The cell membrane isn't just the covering around the cell and its organelles. It is a super important part of the cell that is responsible for controlling what gets into the cell and what can leave the cell. It is described as a fluid because it is quite flexible. It has a complex structure with various proteins and carbohydrate molecules embedded in specific locations that are important to the cell's function and its ability to join other cells, communicate with other cells, etc. Some proteins form channels that let some substances come into the cell and some substances get out of the cell. It carefully keeps some substances from entering the cell. The membrane is made up of two layers of long chains of molecules called phospholipids. Cell membrane, a closer look. This is what a phospholipid looks like. It has one part called the hydrophilic head, which means it likes to be surrounded by water. This head is made up of a chemical group called a phosphate. This is where the term phospho comes from. The second part of it is a hydrophobic tail, which means it does not like to be surrounded by water. This tail is made up of fatty acids, and this is where the term lipid comes from. A lipid is a substance that does not like to be surrounded by water. Importantly, the properties of phospholipid molecules allow them to spontaneously form a double-layered membrane. The cell membrane is made up of phospholipid bilayer. Of the membrane, and the lipid part of the two layers face the inside of the cell membrane. Thank you! I hope you like this informative presentation on the topic of the cell structure. Now, if you recall, we talked about the genetic material known as the DNA earlier during the nucleus. Now, Kavya, next up, will talk more in depth about DNA.
DNA and DNA application. What is DNA? DNA is short for deoxyribonucleic acid. DNA is the material that makes up our chromosomes and stores our genetic information. It is a blueprint for the cell and what it's supposed to do. DNA is what gives everyone their different traits: blue eyes, brown hair, etc. DNA is what makes you, you. The deoxyribo part refers to the name of the sugar that is in DNA, which is deoxyribose. DNA is basically a very long chain of nucleotides. The smallest DNA molecule has over 20 million nucleotides. What are nucleotides? Nucleotides are made up of three main parts: a phosphate group, a 5-carbon sugar, which is deoxyribose in DNA, and a nitrogen-containing base. The difference between nucleotides that make up DNA are the bases. 4 bases are found in DNA. These bases are adenine, guanine, thymine, and cytosine. Adenine and guanine always pair up, and the same thing with guanine and cytosine. Bases that always pair up are called base pairs. The bases are divided into 2 groups, the pyrimidines and the purines. The bases are put in categories based on their chemical structure. Pyrimidines include thymine, cytosine, and uracil. Uracil replaces thymine in RNA. Purines include adenine and guanine. How do nucleotides make up DNA? There are only 4 bases for these nucleotides, but they actually make up all of your DNA. These 4 bases can be put into many different orders. There are millions of different nucleotides in a chromosome, which is a long strand of DNA, making many different combinations possible, which makes everyone's DNA different, and that's why you don't look like anyone else. DNA replication. The human body makes billions of new cells every day. To do this, it has to make many copies of its DNA. This is called DNA replication. Before the cell divides into more cells, it first has to copy the genetic information in its nucleus. That way, the copy of the cell is exactly the same as the original cell. The cell takes about 6 to 8 hours to copy all of the genetic information. Since the cells have to make copies very fast, the DNA replication happens at many different parts of the chromosome. When the DNA is being replicated, the 2 strands of DNA are pulled apart and copied in different directions, so it looks kind of like a fork. This happens at the rate of about 50 nucleotides a second. How does DNA replication happen? The DNA replication is exactly what it sounds like. It is making copies, or replicating, the DNA. It happens when the cell has to make a copy of itself. To make a copy of itself, it has to copy its DNA. The first thing that has to happen in DNA replication is that the double helix shape, which is the shape that the DNA makes, has to be separated. This looks kind of like a zipper being unzipped. The hydrogen bonds between adenine and thymine are broken, and the same thing happens with the guanine and cytosine. Hydrogen bonds are what hold the bases together. This breaks the original DNA into 2 parts, making it look like a fork or a Y. Now, these 2 pieces of DNA can each become new strands of DNA. DNA replication is called semi-conservative. It is called this because half the DNA is part of the old or original DNA strand, and the other half is made brand new. So here's some information on DNA. DNA never leaves the nucleus. Instead, it's transcribed, or copied, into RNA molecules, which may then travel out the nucleus. For example, if the cell needs a protein to be made, then the portion of the DNA that has information on how to make that protein, the gene for that protein, gets transcribed, and the information is sent out the nucleus encoded in a molecule called the messenger RNA or mRNA. Cyril will speak a little more about the major shapes of RNA and their functions. The nucleotides, consisting of nitrogen bases, the sugar and phosphate groups, are brought together in a specific manner. involving specific locations of attachment on the sugar and phosphate molecules, so that the base pairs facing each other are held together in a specific manner. Now watch a search simulation on the pairing of the nucleotide bases in the formation of the double-stranded DNA molecule. So, now that you've seen that, I've just talked about DNA, and next we will talk about another nucleotide, which is RNA. Thank you.
Thank you, Ashwita and Kavya. Now that we've heard about the cell structure and DNA, Siri Chekkarur will talk about RNA, which will be followed by Vigneshwari Shivakumar, who will tell us about making protein using messenger RNA.
So, what is RNA? We just heard Kavya's presentation, so let's start from there. One difference between DNA and RNA is that the sugar is found in the nucleotide. We saw that the sugar in DNA is deoxyribose, and that the sugar in RNA is ribose. The nitrogen-containing bases found in RNA are uracil, cytosine, adenine, and guanine. These nitrogen bases also pair up specifically as AU and CG. We learned that DNA never leaves the nucleus. Instead, it's transcribed or copied into RNA molecules, which then may travel out of the nucleus. RNA plays a key role as an intermediate between the genetic information in the DNA and proteins. There are 3 types of RNA: ribosomal RNA, messenger RNA, and transfer RNA. Ribosomal RNAs are found in the ribosomes and account for 80% of the total RNA present in the cell. Our RNAs leave the nucleus and combine with proteins and enzymes in the cytoplasm to form ribosomes. Ribosomes are the site where protein are made or synthesized. Ribosomes are composed of a large subunit or a small subunit, each of which is made up of its own specific rRNA molecules. Ribosomes may be free, floating around in the cytoplasm, or might be bound to the endoplasmic reticulum. They also might be bound to the outside of the nuclear envelope. The DNA gene sequence is rewritten into an RNA, and then some processing happens to make this a messenger RNA. This is called transcription, and it happens in the nucleus. That's where the mRNA is constructed. mRNA accounts for just 5% of the total RNA in the cell. It carries complementary genetic code copied from DNA during transcription in the form of triplets of nucleotides called codons. Ribosomes get their orders for protein synthesis from the nucleus, where portions of DNA are transcribed to make messenger RNAs. mRNA then travels to the ribosome, which uses the information contained in the mRNA to build a protein with a specific amino acid sequence. This process is called translation. As we saw, genetic code is copied in the form of codons. Each codon specifies a particular amino acid, though one amino acid may be coded for by many different codons. There are 64 possible codons or triplet bases in the genetic code, but only 20 of them represent amino acids. There are also 3 stop codons, which indicate that ribosomes should cease protein generation by translation. Transfer RNA is the smallest of the 3 types of RNA. tRNAs are an essential component of translation, where their main function is the transfer of amino acids during protein synthesis. The transfer RNA brings the amino acids to the ribosomes so that the amino acids can be joined to form the protein. Therefore, they are called tRNAs. Each of the 20 amino acids has a specific tRNA that binds with it and transfers it to the growing polypeptide chain. Proteins are made when the portion of the DNA that codes for that protein is activated. This is called gene expression. Protein synthesis is when information from a gene is read out to make a protein. Translation happens inside ribosomes in the cytoplasm. That's where the mRNA sits between two subunits of the rRNA and a tRNA to which an amino acid is attached. And with a correct 3-nucleotide codon, it moves and sits on the mRNA, delivering the amino acid, which is then attached to a neighboring amino acid. Once this happens, the tRNA leaves. The nucleotides are read in groups of 3 called a codon. Each codon specifies an amino acid. There are 3 stop codons and 1 start codon. So these are the universal codons for amino acids, no matter what the species is. Now Vigneshwari will show a short simulation on how mRNA is used and talk a little bit on how mRNA is used to make a protein. Hope you enjoy!
Namaste everyone. I am Vigneshwari Shivakumar and I'll be talking about how you use the mRNA to make the protein in a simulation. So, the nucleus of your cells contain DNA, which have the genetic code for proteins. However, our DNA cannot come out of the nucleus to help make a protein. Instead, it needs a messenger who can carry its information to the ribosomes so that the proteins can be correctly made. As we discussed before, mRNAs are only made when the body or cell needs a protein. So DNA is transcribed, a messenger RNA is formed. After transcription, the mRNA comes out of the nucleus. The mRNA containing the message from the DNA must be translated so that we can put together corresponding amino acids in the correct sequence, making the protein. We saw that the tRNA containing codons and the appropriate amino acid attaches. The process continues until the ribosome meets the stop codon, and thus forming a protein with amino acids in a specific sequence. So, just to recap, proteins are made based on your DNA. DNAs are long strands of nucleotides, and only certain sections of the DNA are expressed to make a certain protein. This is called gene expression, because only certain areas of your DNA expresses for a certain protein. The mRNA consists of codons, which have 3 bases each. Each codon codes for a specific amino acid. Therefore, the amino acids start arranging when the start codon, which is AUG, is there, and then stops when it reaches the stop codon, and there's 3 of these. The universal set of codons was included in series presentation, so based on that universal set of codons, we can see which codon codes for which amino acid. So, I'm going to be coding this protein called this oxytocin. This has 9 amino acids, and its codons are listed here, and now this is its mRNA sequence. Now I'm going to go to the simulation and show how I transcribe this into the protein. So here's my mRNA, and now it's going to get transcribed. So you can see each tRNA coming and attaching and leaving its amino acid. And now as each tRNA attaches with each amino acid according to each codon, our protein is formed. So this is oxytocin, and by using the mRNA, we were able to construct this protein. Thank you. Now, Aruna will be talking about viruses.
Thank you, Siri and Vigneshwari. Next, Aruna will talk about virus hackers of the cells, and Pranati will focus on the immune system.
Viruses, Hackers of the Cell by Aruna Vaidilingam. A brief presentation revolving around the fatal methods viruses utilize to attack our bodies. Introduction to viruses. So, what is a virus? Well, a virus is a pathogen, a microscopic infectious agent that relies on the host cells to replicate or reproduce. However, during the process of hacking into our cells, these pathogens could implement acute damage on our bodies and kill the cell. This mere nanoscopic particle that may emerge to be harmless could potentially alter your human genome and wipe out an entire bacterium in just an hour. In this presentation, we'll delve into the structure, entries, and viral mechanisms used specifically by the SARS-CoV-2 virus to launch the most dreaded attack of our century, the COVID-19 pandemic. Structure. All viruses are composed of the same components: genetic information within some protein casing. Now, the protein shell that encases the genome is called the capsid, and it's composed of individual units called capsimeres. You see, viruses don't have complex organelles like ribosomes or mitochondria. However, certain viruses, including the SARS-CoV-2 virus, contains a lipid membrane and a viral envelope which wraps all the components together. On the viral envelope of the SARS-CoV-2 virus are spike proteins. So what's so special about these spike proteins? Well, spike proteins are glycoproteins, and these proteins have sugar molecules attached to the strand and are used by viruses to bind to the proteins on the host cell and enter it. Secondly, the viral genome of the SARS-CoV-2 virus is composed of positive-sense single-stranded RNA. This means that the genetic information can be directly translated to produce more proteins. Now, this genetic information combined with the protein casing creates the nucleocapsid. Proteins of the SARS-CoV-2 virus. The SARS-CoV-2 virus is composed of 4 structural proteins, which contribute to shape and movement. These are the nucleocapsid protein, membrane protein, spike protein, and envelope protein. The SARS-CoV-2 virus also contains 16 non-structural proteins: NSP1, NSP2, NSP3, all the way until NSP16. These proteins have diverse roles, ranging from manipulating pathways that regulate the host cell's survival to interfering with the ribosome's ability to make proteins from the host cell's mRNA. Now, let's dive closer to the spike proteins covering their outer membrane. In the process of binding, the S protein binds to a receptor on the cell surface called the angiotensin-converting enzyme 2. Now, the bond between the spike protein and this enzyme is particularly strong, which is one of the strategic aspects behind the virus that contribute to its success. So, how exactly does the virus enter the cell from here? Well, the spike protein is composed of 2 functional subunits, S1 and S2 segments. S1 binds to the ACE2 receptor of the host cell, while S2 fuses the membranes of the virus and host cell. These 2 segments, however, need to be split at 2 sites to expose the viral fusion peptides and actually end up entering the cell. Direct fusion is this process used by envelope viruses like the SARS-CoV-2 virus, in which they merge or fuse with the membrane of the host. We're already aware that the S1 and S2 segments need to be cut to trigger the fusion. So how exactly does this occur? Well, the site where they're cut is called the S1-S2 protease cleavage site. Now, a protease is a protein that breaks down other proteins. And practically for all coronaviruses, the protease in the host cell splits the spike proteins at these sites. And the protein that is found in our bodies that makes the first cut is called birin. Endocytosis. Now, we saw in Harshvita's presentation that the cell is coated by the cell membrane, and we investigated some aspects revolving around the structure and function of the cell membrane. Now, the cell membrane also controls substances that are allowed in and out of the cell, and a process that the cell membrane uses to bring material into the cell is called endocytosis. Now, in terms of the SARS-CoV-2 virus, the spike protein binds to the ACE2 receptor and fuses with the host cell. This process is known as receptor-mediated endocytosis. And what happens during this process is a chunk of the cell membrane engulfs, wrapping around the substance that binds to the receptor protein, forming a vesicle. Now that the virus is inside the host cell, it utilizes its mechanisms to generate copies of the cell. After the SARS-CoV-2 virus enters the cell, it disassembles, and the proteins and genetic information separate within the cytoplasm of the host cell. The virus then takes advantage of the resources presented in the host cell. That means using ribosomes, nucleic acid, amino acids to translate its genetic information and produce multiple copies of its components. The viral RNA and the protein are produced, and they're reassembled to form new virus particles. In fact, up to hundreds and thousands of virus copies are made, exiting the host cell. When the amount of viruses reaches a maximum capacity within the host cell, this influences the host cell to sometimes rupture or lyse. The host cell becomes terminated, and the copies of the virus are then released and free to attack other cells. Alternatively, copies of the virus can also lead through a process called exocytosis, where a vesicle forms around the exiting virus, merges with the cell membrane, and is expelled. Exocytosis is used by the cell membrane to remove substances that the cell does not want anymore and must immediately remove. Conclusion. So in this presentation, we tackle the apparatus, behavior, and the interactions of the SARS-CoV-2 virus within our human body. Without investigating the operation of this virus, scientists weren't able to establish such revolutionary technology in an attempt to prevent deadly cases of this virus. Information on the SARS-CoV-2 virus remains vital as the COVID-19 pandemic continues to devastate our society. So regardless of whether or not one may contain a concrete position in the field of science, everyone deserves a chance to get a glimpse of this virus behind the scenes. Thank you, and I would like to introduce Pranati next, who will be discussing the immune system in depth.
Namaste everyone, my name is Pranati Bhatt and today I will be covering the immune system in this presentation. I hope you enjoy. So, this presentation will be covering three aspects. The first aspect will be the immune system in general. The second one and the third one will be divided for the two parts of the immune system, the innate immune system and the adaptive immune system. So let us start with an overall information about the immune system. So the immune system consists of cells that help the body overcome infectious diseases. The cells in the immune system help defend against harmful particles from making you sick. These particles can be fungi, parasites, viruses, bacteria, etc. And they are generally called pathogens. And parts of the immune system work to keep the pathogens out of the body. Other parts of the immune system act to weaken it or even destroy the pathogen that has found its way into our body somehow. This is basically an overall information about the immune system. Now we're going to be moving on to the two parts. The innate immune system, which is the first line of defense against the pathogens. This part of the immune system basically attacks the pathogens and really attempts to defeat them. And if suppose they get past that, the adaptive system comes to play. This is the second line of defense. In other words, if the pathogens manage to escape the innate system, this part is always ready to be a second line defense to help weaken or even destroy them. So let us start with the first part, the innate system. The innate system is basically a part of our immune system, and our skin has a covering of epithelial cells that are included, part of this. These cells are a protective layer that forms a physical barrier and prevents many pathogens from entering the body. There are other places in the body where epithelial cells form a protective barrier, such as the nose, the mouth, the respiratory tract, the digestive tract, etc. And here, the epithelial cells have a mucus coating, helping to keep the pathogens away. The mucus also contains chemicals that make it harsh for the pathogens to survive. Think of the acid and its role in keeping pathogens at bay. And acts such as sneezing, coughing, throwing up, basically move the air and liquid around so that the pathogens are kept from attacking the body. Fever is used to make it more difficult for the pathogen to survive in the body since it has such a big amount of temperature that is rising and there's so much heat that's made. Additionally, there are actually good bacteria in the skin, digestive tract, etc. that are also part of the innate immune system. These good bacteria help keep these pathogens from harming the body. Okay, so let's move on by talking about some cells that work as part of the body's innate immunity and a brief description of them. So let us start off with the neutrophils. These basically travel the body using the blood as a transport system and swallow the pathogens they find using phagocytosis. Macrophages are found all over the body, and they also swallow pathogens using phagocytosis as well. After that, they release small proteins called cytokines, which cause inflammation and recruit other immune system cells to the area. And last but not least, natural killer cells, which attach themselves to the pathogens and secrete chemicals to kill the pathogens, lowering the number of pathogens that the immune system is fighting. Now let us move on to the adaptive system. So the adaptive system defends against the pathogens that get past the innate immune system. The adaptive immune system works to stop this infection and to develop a memory of the response to the pathogen. This memory will help in quick response to future infections. Vaccines help to teach the adaptive immune system this immunological memory. Some members of the adaptive immune system are dendritic cells, macrophages, B cells, and moving on, we have dendritic cells. Dendritic cells travel to tissues all over the body using the blood. They also swallow the pathogens that they find using phagocytosis and display pieces of the pathogen on their cell surface. This surface signal is called an antigen, and other immune system cells, such as the T cells, are activated to fight the antigen. Macrophages can also swallow the pathogen and present pieces of it on its cell surface. When this happens, other cells, such as T cells, are activated. And we have the B cells, which also swallow pathogens and display on cell surface. In addition to that, the adaptive immune response is to increase the number of B cells. One of the functions of the B cells is to make the antibodies fight the antigen. Moving on, B cells use exocytosis to secrete these antibodies, which are potent weapons against the pathogens because they are tailor-made to neutralize the pathogen. Many of the B cells die soon after this. A small number remain, and they are called memory cells. They can effectively destroy the pathogen if a later infection happens. T cells are moderators of the adaptive response. They basically just set a pace and make sure that there are enough number of fighters are made available. There are the helper T cells, cytotoxic C cells, regulatory T cells, and much more. We also have the helper T cells, which oversee cytokines, which act as a B cell activators. They also increase the efficiency of other immune cells, such as macrophages. We have cytotoxic cells, which are important in viral infections. They kill cells that have been infected by the viruses. Regulatory T cells regulate the immune response. They signal for increased activity early in an infection. They signal for a decrease in the response as the infection is brought under control. Thank you for staying till the end, and I really hope that you learned something from this presentation. I hope you got to know more about the immune system and how it affects your body and what role it plays in your life. Thank you so much. Vigneshwari will now be combining some of the science presented today and to outline some key facts about the mRNA vaccine. I really hope that you enjoy. Thank you and namaste.
Thank you, Aruna and Pranati. Next, we will have Vigneshwari present on the mRNA vaccine, which will be followed by Hriday Solanki and Yukti Solanki, who will give an overview of the mRNA vaccine development. After these two student presentations, we would like to welcome Mrs. Prema Desai to provide a thank you note. Mrs. Prema Desai is the administrator of the Ganeša Pãtašala and a key member of the Pãtašala committee.
So now that we've talked about the virus and our immune system, let us talk about the mRNA vaccine. So, ever wondered how vaccines are made? Vaccines have been made by growing the virus using various processes. Large amounts of virus are made and then harvested. The virus is then used to make vaccines by several methods. First method is to kill it so that we can inactivate it. The virus is killed with a chemical, so the virus is no longer infectious. We've used this method in hepatitis A, polio, some influenza vaccines. The second method is to make the virus less dangerous by weakening it. Weakening means that the virus cannot reproduce as quickly as it normally can. For example, we use this in the measles, chickenpox, and many other vaccines. The third method is to use part of the virus, a protein, and purify it. And we've used this in the hepatitis B, shingles, and other vaccines. So viruses have to be grown, harvested, purified, and treated so that they are appropriate for vaccines, but these methods take time. It takes time to grow the virus in some other organism or cell. Then the virus has to be isolated and purified to medical grade so that it is high enough in quality to be used as a drug. So, we use the mRNA vaccine in order to solve this. The technology behind the mRNA vaccine uses the ability of the host cell, our cells, to make proteins from the codes found in the portion of the virus's mRNA. Then, the newly made protein pieces are used to train our immune system to build a tailor-made defense against the virus. This technology has been actively studied by scientists since the 1990s. So, what is this mRNA technology even about? So let's just look at some of the points that we've learned so far. Known spike proteins of COVID virus are considered the way the virus enters the host cell. We also know that spike protein was identified as the way the virus invades the host cell, because we've had similar experience with the SARS and the MERS. They all belong to the coronavirus family, so we know how it feels. The amino acid specifically, and the specific way that they come together to form the virus's spike protein is also known. The universal genetic codons which code for each of the amino acids is also known. mRNA can be made in the lab using the universal codons because we know the sequence of the amino acids that make up the specific spike protein. Our cells make the spike protein from the code in the mRNA. The spike protein itself is not harmful. It is this outer coating of the virus. The body then recognizes this new protein as foreign, and then the adaptive system will design a targeted antibody to destroy the virus. So making antibodies against the spike protein will give the adaptive immune system the ammunition it needs to fight the virus. But there are some issues with this mRNA. mRNA is unstable. Well, it should be because you don't want your body to keep making the same protein. This is true for any mRNA coding, for any protein in your body. Plus, in a vaccine, it is injected into the muscle and the cells there can break it down. If the breakdown happens, then you won't have enough of the mRNA to make enough of the viral spike protein to generate an immune response. And plus, the human immune system does not like foreign mRNA. This is reasonably because an actual virus can degrade and its mRNA can be floating around. This mRNA is foreign to the host, and the immune system recognizes it as a pathogen, which can cause an inflammation reaction. That could be dangerous. So we did something which is modified mRNA. Modified mRNA is used so that the body's immune system does not treat it as being foreign and does not break it down. This keeps the chances of inflammation response to the mRNA very low. So to protect the modified mRNA from being broken down as it makes its way from the muscle where it is injected into the cytoplasm of the cells where it needs to go, it is wrapped in lipids. We learned that in cell structure, similar to the lipid bilayer that is wrapping the cell. It is this lipid bilayer that the mRNA must also cross to get into the cytoplasm. So taking a cue from nature, a lipid nanoparticle, consisting of a lipid bubble with the mRNA inside, it is made. So the lipid bubble protects the mRNA as it travels to the cells of the body, and the lipid bubble keeps and helps the mRNA cross the cell membrane and into the cytoplasm where the ribosomes are. Very neat, isn't it? The mRNA gets into the cell so that the cell's ribosomes can go to work and make the spike protein that it codes for. The mRNA has to cross the cell membrane to do that, and the lipid nanoparticle helps it do that. The lipid covering helps the nanoparticle with its precious mRNA cargo across the cell membrane, which is also made of lipids. The lipid nanoparticle and the modified mRNA inside of it have to be kept at very low temperatures to prevent them from degrading. Once the vaccine is thawed, it must be used quickly. So, a summary. The modified mRNA, which codes for the spike protein of the SARS-CoV-2 virus, gets into the cytoplasm of the host cell and reaches the ribosomes. Ribosomes then make lots of the pure form of the spike protein because that's what ribosomes do so well. They make good quality protein from the mRNA strand that codes for the protein. The newly made proteins are then displayed on the cell's surface of the immune system to recognize and to tailor-make a response to. All of this uses the host cell's machinery to make a copy of a portion of the virus and the host immune system's machinery to make antibodies to that specific virus. Very cool, isn't it? The coronavirus pandemic has been a very heartbreaking and stressful experience for all of us. Yet we can stop the spread and make the virus less infectious by getting vaccinated. By getting the vaccine, we not only protect ourselves but others around us. When we get infected by the virus, our bodies will already have the antibodies ready to destroy it, and thus we won't be infecting others. With the vaccine out, people getting vaccinated, and us fighting back the virus, our days of returning back to those normal days will be near. Thank you, and I hope you enjoyed this presentation. Next, Hriday and Yukti will be talking about the history of the mRNA technology.
Hi, my name is Yukti, and I'm going to be talking about the scientific history of the mRNA with myself and my brother. Early work. The story of a custom-made mRNA therapy spans 3 decades and started with the work of a scientist named Katalin Karikó. She was a researcher and professor at the University of Pennsylvania. Throughout much of the 1990s, her research efforts were rejected for funding by government and private groups. Even her colleagues considered her work as being not mainstream enough to work. After 6 years as faculty at University of Pennsylvania, she found herself being demoted because her research did not get any funding. She did not give up. Modified mRNA. Dr. Karikó and her longtime collaborator, Dr. Wiesman at Boston University, developed means to tweak the mRNA so that the immune system does not recognize this hybrid mRNA as a threat. This work was published in a series of scientific journals beginning in 2005. Birth of Moderna. Although bypassed by many, it was recognized by 2 scientists, Dr. Derek Rossi and Dr. Timothy Springer. Both were at Harvard Medical School. Dr. Rossi was more focused on using the hybrid mRNA in his stem cell research. Dr. Springer was a professor and an entrepreneur. The story goes on. In 2010, Dr. Rossi, Dr. Springer, and 2 other physicians, researchers from Harvard formed the firm Moderna. This name was arrived at by combining the mRNA and the modified nature of the hybrid version, thus resulting in the name Moderna. Interest in mRNA for cancer therapy. Meanwhile, in Germany, 2 other researchers, Dr. Ugur Sahin and Dr. Özlem Türeci, co-founded another biotech firm. Their interest was not in vaccines, but in personalized immunotherapies for cancer patients. They recognized the vast potential of the mRNA technology. Additionally, they persuaded twin entrepreneur brothers, Thomas and Andreas Strungman, to spin off a new company focused on the use of mRNA technology for cancer therapies. Birth of BioNTech. This new biotech company, BioNTech, was formed. The name BioNTech is also a blended form of biopharmaceutical new technology. BioNTech is headquartered in Cambridge, Massachusetts. Dr. Sahin is the CEO and Dr. Türeci is the Chief Medical Officer. Dr. Karikó and BioNTech. Both Moderna and BioNTech licensed mRNA technologies developed by Dr. Karikó and Dr. Wiesman. Dr. Karikó was hired by BioNTech as a senior vice president overseeing the mRNA work.
Hi, my name is Hirde, and I'll be continuing. BioNTech firms pursue mRNA therapies. Without any public data sharing, both Moderna and BioNTech raised billions of dollars from investors and pharmaceutical companies. Both BioNTech firms worked on developing mRNA medicines that could be safely dosed over and over. Wuhan, China. In December 2019, the International Society for Infectious Diseases, a nonprofit organization based in Massachusetts, sounded the alarm about 11 million people in Wuhan, China being diagnosed with an unknown form of pneumonia. Many patients in China who were diagnosed with this had visited the Wuhan South China seafood market. Work on mRNA vaccines. On January 10, 2020, Chinese scientists posted online the genetic sequence of the new virus. mRNA therapies use the published code to generate a section of virus S protein to be used in the vaccine development. Race for vaccines. Moderna and BioNTech focused on vaccines development using mRNA technologies. Dozens of others were also focused on vaccine development, but largely using more traditional methods, which are more time-consuming. That is, growing the virus, weakening it, or extracting protein from it, purifying it, and etc. Both BioNTech and Moderna started late-stage clinical testing in late July 2020. Pfizer, BioNTech, and Moderna were able to produce mRNA vaccines so quickly because both firms had invested in mRNA technologies long before COVID-19 infections began. These are the references.
Namaste everyone. On behalf of our VALLABHA Ganapati Devasthanam, I take this opportunity to thank Dr. Malini Ravi for her contribution to the Ganeša Pãtašala, teaching our students science and also serving in the Pãtašala committee. Dr. Malini Ravi is a professor in the chemistry department at Lehman's College. She has worked tirelessly in guiding our students in their research on various projects like COVID-19, the wildfires of California and its effects on the environment, which they have presented on a Temple Media. Most importantly, I thank everyone for watching our students' presentation today on the COVID-19 vaccine. Thank you.
Thank you, Brahmanti, and of course the students of the Ganeša Pãtašala, and very importantly our audience on Facebook and YouTube. We would also like to inform you that the Ganeša Pãtašala registration is still open till 31st December. For more details, visit the Pãtašala website. Also would like to inform you of our violin concert that's going to be held tomorrow in conjunction with the Deepavali celebrations. We will have violinist Rina Deepenathur will be accompanied on the Mridangam by Indalur Adarsh. The program is tomorrow afternoon. It should begin at noon. Please do join us on Facebook and YouTube. Namaskaram.